everybody. On to our next talk by David Foster, who's here at Berkeley. Play cell sequences in the hippocampus. Seems familiar.
All right. Thank you, Butler. Yes, it's great to be here at this conference, and let's dive in. So I think I'm going to have some things to say that will potentially mesh or jar with previous ideas that we've heard so far, and possibly ideas that we'll hear afterwards. So that's great. That's exactly why we're here. Let me provide a sort of 5-minute or less kind of intuition to how I'm thinking about what replay sequences are about. So I'm going to tell you about this phenomenon, replay. Quick show of hands, who's heard of replay and knows a little bit about what it is? That's most people. So I think it's okay to do it this way around, but we'll of course explain what it is later. So imagine you're a rat exploring a maze, and you're a Tolmanian rat. So Edward Tolman, who in the 30s and 40s was at Berkeley, actually all the way through to the 60s, and he famously hypothesized that the rat builds a cognitive map of the environment. And this was a reaction against the very simple stimulus response models that were current at the time. So our rat is slowly building up this map as he explores this environment, right? And the point of here is that he can do this very quickly, meaning he only has to experience a transition here, a movement once, to be able to know that that's a possible place he can go. So we sort of think of this model learning as fast to learn, right, in that sense. But now he eventually builds up the whole model, and now he's in the maze, and the question is, what does he do? Where does he go? Which way should he go to get out? Up. Up. Okay. That latency was about 3 seconds. It's a little complicated because you've got to read the map. And what your eyes do is they scan ahead and try and figure out. And there are more efficient algorithms, but whichever way you do it, there's still a problem to be solved, even if you have a map. So in that sense, models can be slow to use. And this was the famous criticism that Tolman was subjected to, although albeit before any kind of mathematical formalisms, which was that his map left his rats buried in thought, right? And of course, that's a bad thing if you're running away from a predator. The other alternative, so classically distinguished alternative approach, is what's called model-free. Here the idea is that you make multiple samples of actual behaviors, and you can slowly build up some notion of how valuable all of the places in this environment is, one trial at a time. So you find some food, and then you know that this, and don't worry too much about the specific representation. There are different ways in which it can be equivalent. But you find that this location is important because it was followed by food. It's one step away from the food, and then another trial you come in and you realize that this location is close to this location, so you can move the reward information back a little bit, and so on and so on. And eventually you can end up with a representation that has some expectation of how far away you are from the goal, but it takes many, many, many trials, to converge, right? So it's slow to learn. But it's fast to use because it gives you exactly the local information that you want. So when you're in a particular position, you know locally which is the step that takes you one step closer to the goal. So this idea has been captured as a trade-off, that animals may want to trade off these two kinds of learning, model-based and model-free. For example, Nathaniel Dorr has talked about this.
Excuse me. Why do you call the second one model-free? I mean, there's still a model, right?
Well,
or is this the sequence?
It would be a very impoverished model where you basically just know, you don't even have a sequence. You have a representation of each location, and with that representation you can associate something, such as an expectation and a scalar quantity like value. So actually this model-free doesn't know anything about the shape of this. It just learns through trial and error and builds up this value function.
You had the shape.
So it's usually thought of as a trade-off, but in fact what you'd really like to do is encode your experiences with the model and make your decisions with a model-free system. Because exploration is expensive, right? And indecision is expensive. So the question is, can you do this? And it's almost a corollary of the fact that exploration is expensive and indecision is expensive, that therefore animals don't spend a lot of time doing those activities, in which case they must have a lot of time doing neither. And we think of these as offline states, rest states where the animals are basically not subjecting themselves to the danger of being eaten. And there's so much time that maybe there's some process you can use where your model can teach a model-free system and you can learn what to do. And that is essentially the way I'm thinking about replay.
So is learning a value function easier when you have a model of the environment? I thought it was easier.
When you have a model of the environment, but you still have to learn it, you still have to derive those values.
So if you have the model environment, you have already the map, right?
Let's say you have a transition function. And the way to think about this is that this is actually a very small, impoverished transition function for the actual decision space, state space that an animal faces in his environment. So the actual real-world goals of an animal, and these are not just locations, but other states that the animal has to make decisions in, there's a very, very large state space that has to be searched through. And even if you use efficient algorithms like value iteration, it's still going to take some time. What you'd like to have is the values right there locally and the actions ready to go. So this is how I'm thinking about it. So let's talk about the data. You've already heard about these cells, the CA1 pyramidal cells in the hippocampus. And I'm not going to introduce this, but we're recording in freely moving animals, and we're isolating extracellularly action potentials from large numbers of cells simultaneously. And I probably don't need to introduce this, but this is a typical hippocampal place cell. So what you're looking at is an animal running up and down a track that's about 2 meters long, sweeping back and forth. This is about 20 minutes of time. This is his position. The gray is the location. He sweeps and then stops and sweeps and stops. He's actually eating something at either end. And on top you see this hippocampal place cell fires in one location. Okay, so that's you can capture that as a one-dimensional place field. Across a number of these cells recorded simultaneously, you typically find that different cells do the same thing but in different locations. So that's in that sense that we have a map of the environment.
Are all the directions selected?
What's that?
Are they all directions selected?
Yeah. So on the linear track, many of these will be direction selective, and each cell to a different degree. But when we do population decoding, we can very clearly pick out the difference between one direction and another, as well as the position. So a few things about place cells, just to contrast them, although this is not necessarily the focus of this session, but compared to something like grid cells, they're spatially stable, right? They do the same thing every time in the same place, and they cover the intervening space. So this is like a hierarchy in my mind of spatial characteristics. Spatially stable would also be true for any representations that are anchored to. I mean, if the animal touches something in the environment that's fairly fixed, that's a spatially stable allocentric signal. But this way he has them everywhere, right? So that's sort of why this is so useful, including places where there wouldn't be any local cues to define those locations. But they're not particularly geometric. This has sort of been a long-standing theme in asking how spatial these cells are. Now with grid cells we have these very compelling sort of geometrical representations. But these come in all sorts of different shapes and sizes. So quickly getting to...
So not being particularly geometric, you mean they don't have a particular shape?
They don't have a particular shape, and it's not obvious how you would derive quantities like metric quantities like distance from them.
Sorry, I have a question. So about your notion of model-free and model-based.
Yes.
So isn't it true that for any model-based approach that you go, you can just build something model-free out of it? So you can just do the learning once and then that's it, right?
Do the learning once, that's
So-
it.
so
Yes, that's what I'm going to do.
let's say you have a model, right, of the environment, right?
Yeah. Let me show you how that works. Potentially. So here's the animal running from one end of the track to the other, okay, and then being stationary. This is a minute now, okay? So this is like a few seconds, and then he's stationary at one position. And these are the 19 cells firing in their classical place fields, right? But what you can see here is while the animal's stationary, there's all this other activity going on. And, just to jump to the punchline here, this activity, if we just zoom in the x-axis, this activity is structured. In this case, it's what we call a reverse replay because the cells that fire in the current location fire first, but then all the other cells all the way back to the beginning of the track. And this is a couple hundred milliseconds, condensing this 2 or 3 second experience. But the thing is, this is happening many, many, many times. And let me just, the 12 strongest events here are these. Okay, so you can see there's actually there's 14, I'm sorry. There's very distinct and repeatable patterns. So if we go back, I just want to make a few points about this. This activity is actually also structured because it's happening during a theta rhythm. So in each theta cycle, you just get a few spikes from each cell. So the sort of local environment for these cells in terms of spiking looks very similar here to here. Here the cells are firing in little bursts of 1, 2, or 3 spikes, and within tens of milliseconds to other cells that represent adjacent positions. And the same thing is true over here. So, but there's a lot more of these, right? So there might be 5 or 6 theta cycles here, but this depends on how long the animal stays here. And in this particular task, he's staying here for a minute and then he's turning around of his own volition. But in general, and if I'm right about just my hunch about exploration being expensive, in general, rats would have very long periods of time where they would be just sitting around, not engaging in activity. And they have all this time to do processing. And the point here I want to make is that this processing is happening. And these highly structured offline runs-through of trajectories are happening.
David, I might have missed it, but can you clarify, what is the blue trace?
Yeah, and so I didn't mention it because I'm not going to say much in this talk, but this is the local EEG, which is also being recorded. In fact, this is one of these channels just filtered differently to pick up rhythms and the local field potential, right? So this is filtered for the ripple frequency of 100 to 250 hertz. So these events happen during these so-called sharp wave ripple events. That's all it says. So reverse replay, was perhaps a bit counterintuitive, but we had come up with this model just to make sense of it, which is that you could actually use this to learn some kind of value function. So don't take this part too seriously, because there could be many different circuits that do it. But the key point is, if you have some kind of fast onset, slowly decaying reward signal, then if it's happening at the same time as a reverse replay, then you're pairing, meaning co-occurring, you're pairing these cells that fire close to the reward with very high levels of this signal, and less as you go back to the beginning of the track. And these pulses of dopamine seem to could potentially have the right time scale for this. So the consequence of this would be that place cells would then acquire this kind of gradient association. We very indirectly found some evidence to support that. So let me explain. This is a busy slide, so I'm not going to talk about it all. This is the slide I used to say that this kind of funny reverse replay was replicated by other people. But I want to focus on this. So this is Dieber and Buzaki, who showed that, in fact, and this is the other way around from the way I showed it, sorry. But here you have the classic place field activity of a bunch of cells as the animal traverses a track. But just afterwards, you get the reverse replay. And just before the animal sets off, you get a forwards replay. So what I want to explain to you here is that because of the directionality of cells that was mentioned before, different cells are firing in each direction of the track. This period during which you get forwards replay of this direction, you're also going to get reverse replay of the other direction, right? That's the flip side of this. If we imagine this same figure for the animal moving the other direction, you'd have both of these. So that's the idea, actually, and we verified that. So during stopping periods, you actually have forwards and reverse replays intermingled.
David? So you must have this. Is there a reward in this task?
Yes. Yes, in Dieber and Buzaki. So.
But in your task, is there a reward?
Yes.
Okay. So what if no reward? Does replay go away?
Yeah, great. So. going well. Okay, so we did an experiment recently where we had 3 epochs of running on a track, and we either increased the reward at one end during the middle epoch, or we actually get rid of it. I'll talk about this. This is actually a bit more complicated because of behavior. But here it's very simple. The animal runs up and down, continues to run up and down, but at one end, in the middle epoch, each epoch is something like 15 running laps, and then there's a break, and then 15 running laps, and then there's a break. And then in the middle, he's getting 4 times the amount of liquid chocolate. And just to get to the punchline here, that has an effect on shockwave ripple rate, which was actually shown by Lauren Frank a few years ago. It turns out when you look at replay, that effect is also there, but it's only really affecting the reverse replays. So these reverse and forwards are all intermingled, but only the reverse replays are showing an increase in replay rate. And in fact, we found the same thing when we decrease the amount of reward, get rid of the rewards. Only the reverse showed a statistically significant effect. It's complicated by the fact that the behavior of the animal completely changes in this case because he's got no food, he doesn't wait around, so he moves. But we actually found interesting effects at the at the other end. Now becomes sort of more valuable. But in the interest of time, I won't talk about that, just focus on this. But it's showing that reverse replay seems to have a relationship to reward, which is in line with what we predicted the function of reverse replay would be, that it's trying to encode something about the rewards and send that information back along the incoming trajectory. Forwards replay then has some other role that we will explore later.
David, wouldn't it be just as important to update your values when you decrease your reward as when you increase it?
Yeah, absolutely. So it would. So there are changes that occur here. For example, now the relative amount, the relative value you might think of this reward, which is unchanged, has increased. And you actually get an increase of reverse replays, not forwards replays, here. And when you bring back, you reinstate the reward, now these 2 rewards are the same, but you get a relative increase now. This is actually has an increased rate over this. So that, we interpreted that as in line with, again, for experimental reasons perhaps, we're finding increases easier to detect than decreases, but the increases follow some of the properties of the dopamine signal, the prediction error signal, and that there's a menu effect here and a kind of prediction error effect, a history effect here.
David,
so, yeah.
Very quickly, do you guys ever test whether the decrease or increase of these rates, is there anything about these patterns, sharp waves, that changes also? Or do those, all of these events look the same? It's just fewer versus more?
We only looked at it in terms of fewer versus more. I mean, we can, and nothing struck me. Obviously, you can see the events, and nothing struck us as different. What do you have in mind?
I mean, amplitude, frequency, winfulness, and stuff like that. In terms of yellow.
So there are differences that occur in replays, which hopefully I'll get to, but we didn't see that as a function of reward. But it's the kind of thing that we're always collecting data on it, so it might be there. Now, quickly, because of what's previously mentioned, I should mention that we've been very interested in the idea of learning. So are the sequences learned? And so I'm going to go over this very quickly, but we found no evidence for preplay, and that's something that we're very interested in. And so I'm not actually going to talk about the way we addressed preplay. We actually replicated the methods of the preplay papers and found statistical errors in those methods that I can talk about if somebody wants to know about it. I will just show this data though, which is, the best sequences we can find in one session on the track, and the best sequences that you can see, during an hour of sleep beforehand. And now we're using this position estimation decoding technique to sort of see the sequences very clearly across something like 80 place cells recorded simultaneously. But you can see it's very easy to detect sequences as soon as the animal starts running, but you don't see anything beforehand. I'll skip that. So this is particularly interesting because we think we've established that there's no prior sequences, but our earlier results had shown that you can see sequences even after the first lap on a novel track. So if we're right about this, these sequences are forming very rapidly. Now that's exactly what I'd said the model should do, right? You should be able to encode your experience very quickly and then start using that to train other systems to make use of it.
Have you looked at these population events when you put the animal on the track, but before it runs that first lap? You were looking at sleep before that, right? But
Yeah.
in the same behavioral state,
Yeah.
that's relevant to this.
So in this paper, we did actually use that, and there was because it's a novel track, the animal will sit there for like up to 5 minutes as he sort of decides whether he really wants to play along or not. And yeah, there was no sequences at all during those periods. So that was actually in the original paper. I will mention this one other study, which gets to the question I was asking Lawrence at the. Where is he?
Yeah.
So this was a paper my student Xiaoxing Wu did early on in our lab, and we recorded on this T-maze. So the task is nominally an alternation, although the animal's not that familiar with it. This data comes from very early in the training process. But the animal's exploring both sides it's exploring all possible trajectories basically on this track. But we find plentiful replays at each of the stopping points. And we considered that there might be two ways in which these replays are generated, right? There may be different pieces corresponding to the different possible entire trajectories, as if you had pre-existing sequences and you map something to them. Or the neurons representing different positions might literally, in some sense, become stitched together in some actual plastic process that comes to resemble, at some neural level, the track itself. And so we found that there were cells that fired, the cells in general fired, when they fired during the replays, they took part in both of these kinds of replays. And despite the fact that this is just showing the firing rate of 80-something cells during replays that start here and end here, or start here and end here. And there's a blue line and a red line. You probably can't see it very well because they're on top of each other. So the individual cells have very different firing rates. Some cells are firing a lot, some cells a little, but they show no difference between whether they're firing as part of a replay that goes up here or a replay goes up here. And that was generally true, and we could shuffle the identity and show there's no difference in the firing rates. So essentially, the cells are acting in the same way in both of these kinds of sequences. So we think this model is the correct one.
So this is area what?
It's the CA1, vanilla CA1 dorsal.
CA1 and I mean, wouldn't you expect some difference because of the L and R part of it? Or is this data?
Well, this is just from this is the C part of those sequences.
Okay.
Yeah. Yeah. So I know I'm running out of time here. I just want to one more thing, which is, so in more recent work, we looked at the classic linear track again, but with much larger numbers of place cells recorded than had been used before, and I think something like 200 cells. And what you're looking at is a replay event that's being depicted in 2 dimensions, position, using this position decoding, but collapsed in time. So you're not seeing time here. You can imagine that at successive frames of time, we would have had different parts of this replay event showing up, and we've just summed it all together. So the remarkable thing is these gaps, right? So we now have better coverage across this linear track than we've ever had before, and yet there are gaps in the representation. And and I didn't make a movie here, but what you find is the replay will hovers and then jumps, hovers, jumps, hovers, jumps. It has that dynamics. And it's nice because again, Lauren's lab had shown that spiking during these sharp wave ripples can be can modulated by the what's called slow gamma, 25 to 50 hertz gamma rhythm, associated with CA3. Here we showed that that is true, but also this hovering and jumping, the information content is also similarly modulated. So it really is the case that within a roughly 30 millisecond period, you get a hover, then a jump. So what do we make of this? So this now maybe we should take with a grain of salt, but it was an old idea that if you want to study heteroassociatively a bunch of items in a hot field net, this will break down by itself. I mean, I think the idea here is you have a well-stored sequence. But if you have to store fresh and just learn online a bunch of patterns, then generally speaking, it's not a very stable pattern retrieval mechanism. And Hopfield actually noted that in his 1982 paper. What you get the guarantees for with hot field nets is autoassociation, right? They will relax to the attractor state. So it's an old idea, and I think the last time I talked about this, actually a few years ago, Andreas Hertz had something to say about this. Hi.
It's an old idea, but it's wrong.
Yeah, okay, that's what you said. So we won't spend too much time on it. But the idea was that that what you wanted to do was have fast synapses to error correct individual items and slower synapses to move. And then what I think is correct about that is you can store sequences very reliably using that mechanism. That doesn't mean there couldn't be other mechanisms. And then the late John Lisman had proposed this as a model by which CA3 could store sequences during theta. So that's our basic conception, that this dynamics now, which is only available to us because of the number of neurons we recorded at the same time, at millisecond, I've got to make this plug, for electrophysiologically, it's just the case that with calcium imaging right now, all of this is, I love calcium imaging, all of this is not available to us because, this is, the entire replay sequence is happening within 100 or 200 milliseconds, but now this dynamics is happening on the order of tens of milliseconds, right? And we think that within each, we predict that within each slow gamma cycle, you're actually going to have a strengthening of the representation during this hover. So there's information in there which is going to be hard to get by any other means. And the idea was maybe this reflects the circuitry. So I mean, CA3, well, I won't belabor that point because that's interesting. So one of the most robust effects in my lab across everybody's data sets is the progressive change in replay that happens with experience during a session, right? You often see replays, they start off fast and they get slower, which is a little counterintuitive. You might think that if you're increasing the synaptic drive or increasing, something's being learned, that this should get, these sequences should get faster, they should recruit more spikes and so on, but they should get faster, but they get slower. This actually makes perfect sense from the point of view of the value learning that I was talking about right at the beginning, because often in these RL models, you have different needs at the beginning of training and at the end of training. Particularly at the beginning of training, you don't know anything anywhere, and you need to get that information out into the environment, meaning your representations of other places need to learn something fast. And so replay sequence that within a, let's say, fixed synaptic window for downstream systems need to get that information from one part of the environment to the other quickly. Later on, you now need to smooth out the inconsistencies and reduce variance. You care less about bias and more about variance. Now you want a smaller synaptic window. So I can see I'll pick. I'll show two things. One is, so we think that this process of elongating the replays actually corresponds to the strengthening of these attractors. And then I will entirely skip the other part of the talk and give you in 3 seconds my 3-minute what's the biggest question, right? Because that somebody wanted to know. The most pressing question. Replay has been reported in other parts of the brain, yes, but it's still that sort of 1990s hippocampal replay where, there's a bit of a correlation or maybe some hint of a sequence. And now we're seeing in the hippocampus that there's this highly structured activity. It suggests all sorts of complexity and structure that could have all sorts of functions. But the hippocampus is, by a sort of technical accident, the only place I would say that replay has really been looked for at that kind of resolution. It's the only place where people have routinely recorded from hundreds of cells at a millisecond resolution. And if you don't do that, you don't see any of this stuff. So the question for me is the hippocampus a special thing? Is replay a special hippocampus thing that has something to do with memory consolidation or value learning? Or is replay ubiquitous? And every area of the brain that has recurrent circuitry, every part of cortex, and many other parts too, has this kind of rich dynamics, in which case we would have to quite seriously revise many of the ways that we think about information processing in the brain. And I'll thank, well, they're all sitting there. I'll thank my lab.
I'm just going to ask one question because maybe others have the same. I didn't get it. Are you saying, related to the previous discussion that we had, are you saying you provided evidence for or against that these sequences are sort of pre-programmed random sequences that then attach to the external sequences of the world, or they are being trained representations of the actual trajectories of the animals? Which one was your data more consistent with? Because I'm sorry.
Yeah, so the finding as reported of preplay is that there are sequences of neurons firing that come to be mapped onto places subsequently experienced. So that if you form your template out of the experiences, you can go and look previously in time and find those replay-like sequences before the experience was ever took place. And we find no evidence for that, and we found, we think, errors in the original analyses suggesting that has not been demonstrated.
But the absence of pre-play doesn't necessarily mean, maybe the rat just doesn't, when it's not moving, it just isn't pre-playing. But that doesn't seem to rule out the hypothesis that the sequences then, once the animal starts moving, are being generated are kind of these random hash codes. Do you agree that it hasn't ruled that out yet, or?
Well, you can generate a random hash code on the fly, right? But you're suggesting that the power of this is that they're already, imprinted into the system, so that it's right, which is fascinating. But I would say my most extreme statement about that is that I don't believe there's any evidence that stands up that's been shown that occurs. So we're sort of back at 2010. And it might, but it's logically inconsistent with several of the findings that we have that sequences come to quickly represent complicated, T-junctions and other structures, spatial structures, in such a way that after just a few trials, they can be, sequences can be generated from them. So that's my flagpole.
So I'm wondering about the discrete jumps that you observe in replay. From what I think we know about play cells, there's no evidence that the states in the hippocampus are discrete. The play cell coverage seems to be quite smooth. And we also know that it's possible to construct attractor models that have this property, that have a continuum. So do you imagine that discrete states reflect the discreteness in the attractor states available in CA3, or that they reflect some choice in the wiring up of events?
You're asking if they're functional in some way, useful, or?
You're suggesting that the discreteness is reflecting some inherent discreteness in the representation or attractor states available. And I'm not sure that is consistent with other things that we know about representation.
Well, for an experience like this, you can have different replays can use different sets of, let's just call them hovering points. Hover points.
Hovering points can vary.
So they can vary, although there's, actually the full study of this is yet to be done, where we cluster them and show that, there's actually an interesting, probably just a small number of possible sets of hover points. But it's not the case that you're stuck with, the animal's completely stuck with dead zones where you can't replay, and retrieve any information. That doesn't happen. But it's very surprising. I just expected that as we added more and more cells, we'd see more and more smoothness. And that, I would say we have strong evidence that that's not the case. So I don't know quite what to make of it.
Hila?
So, David, I mean, you have conceptualization of these replay events as really value-based learning, but what is the actual experiment that shows that really, planning and value-based learning are based on these sequences, right? I mean, it is the case that, some learning, like Lauren's work on jamming sharply ripples, affects some memory-based decision task or station.
Yeah.
But what is really how do we know that this is what's
Well, I think I guess several of us have been chasing this, but it's a bit elusive, but that's to see the value function itself. Well, you might have been, Lawrence agreed with you, but might have been thinking I was going somewhere else with that. But one of the things is seeing the value function itself, right? There's bits and pieces suggesting that cells in the nucleus accumbens, for example, might ramp up their activity on the approach to a goal, but what I'm talking about, what I seem to be talking about is quite sophisticated value functions, right? Because any given task is going to be quite a bit more complex than just a single approach to a goal. And so, you could say, well, it's very unlikely to have a value function that's so contorted and complex. But the replays are there, and they're pretty contorted and complex. So, but we've been recording in nucleus accumbens. It's difficult, and the responses don't fit that model yet. So we're interested. I think there should be some kind of value function or pieces of a value function, or we should have some notion of how this contributes eventually to decision making. But that's really not understood yet.
Lauren?
Yeah, just following up, I totally agree with everything you said, and my sense is this is a hypothesis. That's all it is right now, right? It's a guide for us to plan our experiments. Yes. The thing I was going to ask about was sort of related and implicit in Hila's question. This is slightly selfish given what I'll be talking about later. Do you mind doing a 10 or 15 second or 30 second summary of the stuff you skipped on future stuff? Because that's sort of also, I think, generally relevant.
We should not have him do that right now.
Okay,
we have a few more questions. And
yeah, I'll just
30 seconds can turn into 10 minutes.
Yeah, well, I won't say anything. This is a video that has
Sorry, I apologize.
real time and sharp wave time stuff happening, so you can watch that and I'll answer.
Jerry?
Yeah. Is there an easy way to relate this to the two-dimensional maps and the simulation story and all that?
A little more to...
I think that might have been my question.
What happens in two dimensions? So actually, this kind of stuff happens in two dimensions. So you get replay sequences that are focused in one location, so they don't fan out. That's kind of interesting. And they often predict or seem to predict what the animal's going to do next. So there's this part of the story that we haven't fully got to grips with is the idea that sometimes these events may actually relate more to planning immediate behavior. And that's where I said reverse replay sequences were for value and they make sense. These forward sequences may also have a different role. But the jury is not out on this, and there is interesting modeling work coming out of Nathaniel Dawes' lab right now suggesting that there may be an integrated perspective where everything is value learning, even the forward sequences. So I'm sorry I didn't really talk about that in more detail, but there's a very rich and interesting set of activity patterns that you get in two dimensions that looks like the animal's thinking about what he's doing.
Okay, next to the last question, Andrea.
Is reverse replaying compatible with the idea proposed by Lawrence of a fixed trajectory or a fixed pattern sequence? Because how do you put then tags in the other order?
Yeah, again, I have only hypotheses, but the way I've been thinking about it recently is that the latest results from the spike time dependent plasticity people say that under various neuromodulatory conditions, you can have perfectly symmetric plasticity. So there's modeling work by, it comes out of Sampolinsky's work, but basically showing that you can do symmetry break. If you've got symmetric connections, you You have some method of, you have a bump on a hill, you can squash the bump and make it move, and you can have, you need some kind of momentum to keep that replay moving. There's ways to make that happen, but basically you can have symmetric connections, forwards and reverse, generated during behavior, and you can have reverse and forwards replays come out of that.
I think my question is much simpler. If there is this underlying sequence that has one direction, as in Lorenz's framework, how can you play it backwards?
Yeah, I'm doing a bad job, but so you do this, right? These are the places, right? What I'm suggesting is, in plasticity terms, you do this, and then you might think that if you're sitting here, you don't go anywhere, but there are ways to squash that bump and make it move. So there are ways to do this. It's models for everything.
I think that might be an incompatibility, though. If a network is fundamentally liking to generate A, B, C, D, E, it probably doesn't like to generate the opposite. I mean, I don't know. Maybe that's just pure speculation. Okay, wait. What?
Okay, I'm trying to think about how this way of thinking would fit with results of inactivations or lesions of hippocampus under different conditions. And so if you had an animal learning under conditions of satiety, for example, you might predict that if you then inactivated hippocampus, you shouldn't be able to use this information to make value-guided decisions to goals. Is that actually what people see?
Okay, devaluation experiment.
Well, you're saying that you're using what you learned in the context of reward to do all the replays, develop a value function over your map, and then use that for later navigation.
Right.
So that would predict that if you learned the map under conditions where you weren't being rewarded, say because you were satiated, right, that you shouldn't be able to use that map for value-guided decisions if you then inactivated the hippocampus, right?
I wouldn't make that. I wouldn't make that prediction because the model. So there's two things, right? There's model learning, and then there's integrating the reward. And I mean, I can just make it up, but I can say model learning can proceed, in the absence of latent learning style. The animal's just sampling transitions, and and do we know this? I'm not sure if we know this though, because I don't really believe mouse replay yet, and all rat replay experiments have some kind of reward in them. But if you can sample the reward, the environment without any reward, I would have no problem believing you can replay it. And then the integration with reward to generate a value function is a sort of a separate thing. But.
Okay, let's thank David.